We talked about earlier is that the flow of information is going to happen from the presynaptic neuron to the postsynaptic neuron, so it's going to flow in this direction. Okay, and one more thing that I want to talk about. Does anyone know how you can increase the speed of the action potential down the length of the axon? Yeah, great. Okay, so if you're taking this action potential and to increase the speed, you can have myelination happen. Okay, and we'll talk about what myelination is here and the cells that do it in the second part of the talk today. And there's one more thing that you can do. Myelination, you wrap it with a myelin sheath, and that's going to help increase the speed of the action potential down the axon. How about the diameter of the axon? Do you think it would be faster if it were bigger or smaller?
Bigger.
Yeah, it's actually bigger. Increase the size of the axon. And the way to think about that is if this were a garden hose and you had a really big garden hose, you get like a gulp of water without a lot of resistance to the flow. But if you had a really narrow hose, you have a lot more resistance to the flow of the water. So you think about electricity and water being, I don't know if you got that in physics or not, but think about electricity like water. There's less resistance to the flow of the water or the electrical signal down the length of the axon if you actually increase the size or the diameter of the axon itself. Okay? Question?
Gabby, what does it say after flow of?
Flow of information. It's not real clear on this, I don't know why. But when I'm Xerox, it'll be better. Flow of information. So, yeah, we talked before about the flow of information happening from a presynaptic neuron to a postsynaptic. So you go from an electrical signal to a chemical signal. Chemical signal is your neurotransmitter. Yes, Hannah?
Does that say increase speed after the action potential?
Yeah, to increase the speed of the action potential, the two things to do would be to myelinate or to increase the diameter. The larger diameter axons have an increased speed of transmission. Josh?
So that says myelination?
Myelination. Mm-hmm. You can say myelin sheath, myelination. We'll talk about that in a minute. Try to understand what that means. Any other questions? Okay, let's move on. We're doing good. Now, thinking about, the next is neuron histology. Okay. I like to talk about, what does histology mean? Does anyone know? Tissues, but so study of tissues, but anything else? Anything else you think about when you think about histology? Yeah, cells, how they look. Like how they look under a microscope. You know, histology is how things look. When you say study of tissues, it's usually microscopic, right? There's usually some cell staining involved, that sort of thing. So we're going to look at different kinds of neurons and what they'd look like if you were looking at under a microscope. Here I have function and I have structure on the side. The reason why I have that is because you will learn that if the neuron looks the same, then it has the same function or a very similar function. And we talked before about, let's keep it green. I wish I had better green. We're going to start over here and we're going to say sensory neurons. And sensory neurons in general, are they afferent or efferent? Afferent. So afferent means information coming into the system. Sensory is afferent information coming into the system. And I have two different categories to think about. Sensory information coming in from the body. What is that called? We talked about that a little bit before. Does anybody remember? Somatosensory. So there's somatosensory information. Okay. So somatosensory information, just general information coming in from the body. I think I locked up. Oh, there it goes. And then the other, we have some special senses that we considered before too. And specifically, we're going to consider vision and olfaction. So neurons involved in the visual system, specifically in the retina, and we'll talk about that when we talk about vision. And olfaction, those neurons that are involved in receiving scents or smells. Okay, we're going to find out that your somatosensory neurons have a very specific structure. Okay, the name of this structure for somatosensory is going to be pseudo-unipolar. So that's P-S-E-U-D-O. Unipolar is U-N-I-P-O-L-A-R. So these are pseudo-unipolar neurons, are your basic somatosensory neurons. Okay, your pseudo-unipolar neurons look kind of like this. Up top they have dendrites. Okay. And coming out dendrites you have an axonic-like structure. And then you have a little stalk. And coming off the stalk you have your cell body. Okay, and then coming back down, another stalk. And then you have your axon terminal. There's your nucleus. So labeling this is your pseudo-unipolar neuron. And these are your dendrites. Are here. Okay. This is going to be your cell body. Okay, and then this is going to be your axon terminal. Okay, and we talked before about the flow of information happening from the dendrites all the way through to the axon terminal. And that little cell body, we talked before, where do, and this is going to be a first order neuron in the somatosensory system. Where did the cell bodies reside in the PNS? Does anybody remember what part of the spinal cord? Going to be, your cell body is going to be in your dorsal root ganglia. We'll talk about that as time goes on. That will make more sense. But this little stalk-like structure with the cell body sticking out, that's going to be in the dorsal root ganglia. The dendrites are going to go out, receive sensory information from the environment, and this portion, the axon terminal, is either going to synapse right in the dorsal gray horn or go right up the spinal cord. We'll talk about the difference of those later. Questions about that? Okay, that's your standard somatosensory neuron. Your somatosensory neurons are going to be your pseudo-unipolar neurons. Now your neurons for vision and olfaction are going to be a little bit different looking. They're similar, but a little bit different. And they're known as bipolar. B-I-P-O-L-A-R. They're bipolar neurons. So both the visual system and olfactory system have bipolar neurons that are receiving sensory information from the environment. The structure of the bipolar neuron is similar in that it has these dendrites sticking up here. Okay, and then stalk-like structure, but I mean the axon-like structure, but this time there's not going to be any stalk. That axon-like structure is going to butt right into the cell body, and then another axon-like structure, and then there's the axon terminal. Okay, so essentially the only difference between these neurons is this is a bipolar. Doesn't have a stalk for the cell body to sit on. It's got the cell body right in the middle of the cell. And as I said, so here's going to be your dendrites. There's your cell body, and there's your axon terminal. Okay, overall, your pseudounipolar neuron is going to be somatosensory, basic somatosensory first order cell neuron sitting in the dorsal root ganglia of the spinal cord. And your bipolar neurons are going to be in your olfactory system, actually in your nasal cavity, and the visual neurons are in the retina. Questions about sensory, afferent information? Let's go ahead and let's divide this. I'll just make another section here with my little. Okay, so we've talked about the sensory neurons, or the afferent neurons. Now we're going to move on to the efferent neurons, which are your motor neurons. Bless you. Your motor is your efferent. Okay, your motor neurons, and like I said, motor neurons, it's a 2-neuron sequence. There's an upper motor neuron and a lower motor neuron. Regardless, they all have the same structure, and these are known as multipolar neurons. So your motor are going to be your multipolar. Okay, your multipolar neurons. And your multipolar neurons are nothing more than your basic neuron structure that you saw with my Suffy, right? They look just like this guy. Okay, not much different. So when we draw that, draw it in red, what you see is you have an area up here where there are dendrites that are coming off of the cell body. Okay, and then off of the cell body you also have a very long axon, and that's going to be characteristic of your motor neurons. They have this very long axon, and then they have their axon terminal. Okay, let's see what that is. So these are going to be your dendrites. And this is a characteristically long axon, specific for the motor system. And then here's your axon terminal. Okay, motor system is really simple. Efferent information coming out is going to be transmitted via multipolar neurons, structure of which is your basic neuron-looking cell, dendrites attached to the cell body, soma, very long axon, and your axon terminal at the bottom. And the last little section we have here is going to be something known as interneurons. That in brown. The last little category are interneurons. And these are nothing more than connector neurons. I'll just put connector. And they're only in the CNS. Well, only in the central nervous system. Essentially, we're going to look at some connector neurons that are more commonly found in the spinal cord, and some that are more commonly found in the area of the brain. But essentially, there's 2 kinds of connector neurons that we need to know about. The first is going to be, I'll make them both brown. The first is going to be your anaxonic. So, bless you, ANAXONIC, your anaxonic neuron. And your anaxonic neuron, just like the word anaxonic might lead you to believe, doesn't have any identifiable axon. Okay, so it essentially looks like kind of the multipolar top. So you've got these dendrites that stick out of a cell body with no obvious axon. One of them actually does function as the axon, but when you look at it, it doesn't, you really can't tell which one is functioning as the axon. Okay, so it's just a basic structure of a cell body with some projections sticking out. Okay, in addition to the anaxonic neuron, you're also going to have another kind of multipolar neuron, but this kind that's an interneuron has a much shorter axon. So this is going to be your multipolar neuron. It's going to look just like the motor neuron, but a real short axon. Here's the axon. Okay. So this guy, essentially the same, just has a short. If it's a connector, it has a short axon. And if it's part of the motor system, it has a long axon. That's it. That's what you need to know with basic neuronal histology. Pseudo-unipolar are you're going to be your somatosensory for the most part, your bipolar, vision, olfaction, and your multipolar part of your motor system. You also have some interneurons, which are nothing more than just connector neurons. They're either anaxonic or multipolar with a short axon. Questions? Pretty straightforward. Nobody here's into histology. Okay, I understand. It's Monday. Alex?
When it comes to the motor and the interneurons, the dendrites, the cell bodies, cell bodies right there.
Yeah. Yeah, that's all the cell bodies, just your basic neuron structure. So your dendrites are coming off your cell body. They kind of look like a palm tree, I guess, so the middle cell body. Any other questions? All right, pretty basic. Now let's move on to glial cells. What I'm going to do for the rest of the talk is talk about some different kinds of glial cells. The majority of the glial cells we're going to talk about reside in the central nervous system or the CNS, but there's just a few that we'll talk about with the PNS. Important for a variety of reasons. What you see here, this is your basic neuron, right? This is going to be your multipolar neuron because here's my cell body with the dendrites, really long axon. Okay. What we're going to talk about first is something called astrocytes. And astrocytes are star-shaped, I'll put it up here. They're star-shaped cells. Okay, star-shaped cells. So glial cells aren't neurons, they're neuronal supporting cells. Okay, they're the other thing that's in your CNS and PNS, aside from your neurons. Okay, what I said before, this is your basic neuron. There's your axon terminal, so this is going to be your basic neuron. We could even say multipolar neuron. Now we know what that is. Okay, and down here what I'm representing is just a blood vessel. So if you want to think about this here as one of your cranial arteries. This is going to be a blood vessel in the CNS. Okay? ACA, MCA, pick your favorite, PCA, all those good ones. Okay, what I'm going to do is I'm going to draw my astrocyte right here, and it's going to be kind of a connector between my neuron, or my multipolar neuron, and my blood vessel, and a few other things. So, like I said, they're star-shaped cells, so I like to represent the star shapes as little feet. So coming off, I'm going to draw like little feet here. Okay, I'm going to have 3 little feet making contact with this neuron, with the neuronal cell body. Okay, so this is going to be my astrocyte. And then I'm going to have 3 little feet coming down here and making contact with the blood vessel. But I want to emphasize that these little feet are, when I draw them, are juxtaposed. I mean, they're really close together. They're creating a tight space. Okay? So there's 3 little feet, and they're all together, sitting on that blood vessel. Okay. And then I'm going to put 2 more little feet up here. There's 1 foot there, just sitting out into the extracellular space. Okay, and astrocytes, and here's the nucleus. Astrocytes are going to be a basic supporting cell. They are the most prominent glial cell in the CNS. Their job is they have a couple of jobs that we're going to talk about. Their basic job is to help support the neurons in the CNS. Let's label some functions here. So there's going to be this first function. If you notice, I put these little feet from the astrocyte really close together. Okay? And that's indicating the very first function that I want to talk about. And that's going to be to help support the blood-brain barrier. So the little feet of the astrocyte are all really close together, and it's going to help prevent things from coming out of the blood supply into the CNS that aren't wanted. Okay, so the first function for the astrocyte is to support the blood-brain barrier. Okay. Everybody good with that? Okay, now the second function I'd like to talk about. Let's go ahead and put it over here. Inside the blood vessel, one of the functions of the blood supply is to provide food to the central nervous system, to the nervous system in general. And food, the brain actually prefers glucose. That's just its preferred food source. So just to make it generic, I'll go ahead and say food. So there's food free floating, or glucose free floating in the blood. Okay, and that's going to be free floating in the blood supply. And what happens is it's the job of the astrocyte to take up that food and deliver it to the soma. That's going to be the second job of the astrocyte. So the first is to help support the blood-brain barrier. The second is to take up food from the blood supply and deliver it to the neurons in the CNS. There's going to be another special job. We talked about coming down this axon. At times there will be an action potential. So, and with that action potential, what was the name of the chemical that is released from the cell that results in hyperpolarization of the neuron? Does anybody remember? Potassium, yeah. So what happens is there's potassium released due to the action potential. Okay, it's going to be the job of the astrocyte to take up that potassium from the extracellular space and help keep it neutral. Okay, so the third function is going to be potassium uptake after an action potential. Okay. It just helps keep that extracellular space more neutral. So the astrocyte just sucks up that extra potassium, gets it out of the way. And there's one more function, we'll put it here. That's a job of the astrocyte, and that's going to be the release of something called neurotrophic factors. Neurotrophic factors. Neurotrophic factors. Does anybody know what those are? They're going to be released by the astrocyte to help guide the axon. So they're important for development, neuronal growth. After a CNS insult, there could be damage to some of the axon and it's not making a proper connection. So during neurorehab, which is what we do, neurotrophic factors released by this astrocyte are going to be responsible for guiding that axon back to make the connection where it's supposed to. Okay, so they're just guiding factors. They're proteins that guide neuronal trajectory. So these are going to be responsible for maintenance of neuronal projections and guiding the axons to where they're supposed to go. Okay, those are the four basic functions of the astrocyte. Again, these are the most predominant glial cells in the CNS. They're going to help support the blood-brain barrier. They're going to help uptake food and deliver it directly to the soma of the neuron. They're going to help uptake extracellular potassium that was released due to the action potential. And they're also going to help release neurotrophic factors, which help guide the axonal trajectory. Questions? That's going to be your astrocyte. That's the big one, star-shaped cell. The second one we're going to talk about is a microglia. A microglia just looks like, it's a small microglia, so small cell, essentially. Going to look like this. Little fringes hanging off the edge. Okay, the word micro is small. So a small glial cell. And the best way I know to show you what they do is to draw this. What does that look like? Not bacteria. Close though. Huh? It's a viral particle. Yeah, with COVID. Those are going to be, I'm just drawing basic viral particles. Kind of think COVID. Okay. So your microglia are going to act as the central nervous system in the CNS. Central nervous system. I'm sorry. They're acting as the immune system of the CNS. Okay, so your microglia, these little guys, are immune system in CNS. And Their job is to seek out and destroy unwanted visitors, like viral particles, bacterial particles, anything that your immune system would want gone. That's what that does. Okay, so 2 basic glial cells to start with. You have your astrocyte, it's predominant, and you have your glial cell. Pretty important to have a good immune system in your CNS. And moving on, we have talked about this one already a little bit. When I talked about the choroid plexus, I'll talk about ependymal cells. Okay, ependymal cells are just another kind of glial cell with a special function. So, little review. These guys are going to be, what are these things here? Does anybody remember? Yeah, those are your thalamus. Representative of the thalamus. Now, we talk about there's a ventricle that lies in between the thalami. Does anybody remember what ventricle this would be? Yeah, that's your third ventricle. So it's going to sit right here. So I think of it like a nose for the 2 eyes. And that third ventricle is here. And then connecting to the third ventricle, there's 2 lateral ventricles. There's 2 little foramen. Remember those? Those are your foramen of Monroe. Okay. Out of your foramen of connecting your third ventricle to your foramen of Monroe is going to be your lateral ventricle. Okay, 2 lateral ventricles here coming out. Okay. And does anyone remember the name of the ventricular system that goes through the midbrain? Yeah, that's your cerebral aqueduct. Quick review. I've always made it like a ladder. Okay, there's your cerebral aqueduct, and then underneath your cerebral aqueduct, you have connected to your 4th ventricle. Here's going to be your 4th ventricle. And your 4th ventricle has these little foramen on the outside. There's 2 that are lateral. Anybody remember those? Yeah. Foramen of Luschka, Luschka, Luschka for lateral. And then there's going to be 1 here in the middle, kind of sticking back. Does anybody remember that one? Yeah. Foramen of Magendie. I remember that because M for middle. M for Magendie. We talked about the importance of those earlier today. And then your ependymal cells. So your ependymal cells, what color am I going to make them? Let's make them kind of this purple. Yeah, red. Okay, so ependymal cells are going to be nothing more than small columnar-shaped cells that line your ventricles. They're going to line all of your ventricles. So you have these cells here. They're columnar, or like a column in shape, and they're responsible for lining the ventricles. And I'm just going to draw them here for the sake of time on this lateral ventricle, but realize that they line all of your ventricles. And these orange are your ependymal cells. They're nothing more than specialized cell type that line your ventricles and line all of your ventricles. And you should also know that they have a little hair or a cilia that comes out of them. Does anyone know why they would have a cilia? What's the purpose of the cilia? Yes, absolutely. So the cilia is going to help by promoting the flow of CSF through the ventricular system. And if you notice, I left a little hole here because I wanted to draw in a few of the cells that are responsible for making CSF. That's too close a color. I'm going to make them. Here's this color here. Okay, and these are going to be the cells. They're a specialized filter kind of cell that's responsible for making CSF. Does anybody remember what those are called? Yeah, this is your choroid plexus. So your choroid plexus, they're nothing more than specialized ependymal cells, and they're going to be responsible for making CSF. Essentially, they filter incoming blood supply coming into the area and promote CSF. Produce CSF. Questions about that? Just a little bit of review. We did a little bit of the similar structures in the starter class today. So just went through and reviewed the ventricular system again. You should know that all of your ventricles are lined with ependymal cells, another kind of glial cell, columnar epithelial cell, essentially with a cilia that helps promote the flow of CSF. And choroid plexus are nothing more than specialized ependymal cells to help filter blood to make CSF. Questions? All right, almost done. Yay. Let's move on. Let's look at the next one, which is going to be the star of the show since we're talking about neuronal conduction. It's going to be your myelinating glial cells. Two different kinds, depending on whether you're in the CNS or the PNS. Does anyone know the name of the cells that are involved with myelinating neuronal axons in the CNS? Those are called your oligodendrocyte. Oligodendrocyte. Okay, these are oligodendrocytes, and they're myelinating glial cells that are only found in the CNS. Okay, I have a good friend who had a cat named oligodendrocyte. She called him Ollie for short. So. Kind of a cool name. Okay, so looking on this side, we're gonna start with the CNS. Okay, and like I said before, here's your dendrite, here's your soma, and this is going to be your axon. So your myelinating glial cells are going to be wrapping themselves around the axon of the neuron in the CNS. So to represent that they're wrapping around, I have this little kind of like a spiral here. Okay, I'm going to draw it down, and that just means it's wrapping around. So these are all actually from one cell. In the CNS, the oligodendrocyte has the ability to have one cell myelate multiple neurons, multiple neuronal axons, as well as multiple sites on a specific axon. Okay, what you have here is these are the myelin sheets that are wrapped around the axon. They have little appendages that come off. And then these just connect to the cell body. So you can see that this one oligodendrocyte is responsible for myelinating 3 different areas of one particular neuronal axon. It could also send out other appendages that are responsible for myelinating other neuronal axons nearby. Okay, so it's not limited. One cell, one oligodendrocyte can wrap the axon in multiple spaces. Everybody see that? Good? Okay. Now, in the PNS, you're going to have, the CNS is oligodendrocyte. Your PNS is a Schwann cell, SCHWANN. It's a Schwann cell. Okay, a Schwann cell is nothing more than the myelinating supporting cell in the peripheral nervous system. Okay, oligodendrocyte, CNS, Schwann cell, PNS, and the Schwann cell looks similar in that it wraps around, but every time it wraps around, one individual cell is responsible for wrapping a small section of the axon. So here's the Schwann cell, and there's the nucleus. So this is the entire cell here is wrapping around. Okay. So these are 3 individual Schwann cells that have wrapped themselves around the axon of a neuron in the peripheral nervous system. Pretty straightforward. And we talked before about the fact that the myelination of the axon is going to help speed the conduction of the action potential down the axon. Talk about that a little bit more on the last slide. But that's their sole purpose in life. Without those myelin sheets, it really slows down the speed of conduction of the action potential. Questions? Okay, moving on. So OIs and Schwanns. And then the last slide is going to be neuronal communication with and without myelin. Okay, just a few important things to talk about here. Okay, so just to keep things simple, I have up here, this is going to be my presynaptic neuron, here's my postsynaptic neuron, right? We talked about this before. So this is my dendrites, my soma, my axon, axon terminal, and then this is going to be my synaptic cleft. And we're looking at the flow of information going in this direction. Okay. Right now I'm just going to start up here in the top, and then I have CNS and PNS to talk about different kinds of myelination disorders that you might come across in your clinical future. But for right now, I'd like to start up here. I'm going to say that this is an axon, or this is a neuron that's in the PNS, because I think the Schwann cells are easier to draw than the oligodendrocytes. So I'm just going to draw in my Schwann cell here. Okay. And then I just have a couple of Schwann cells that have wrapped themselves around the axon of this neuron. Okay. And I'll label those. We talked before about having a cell reach an action potential, and reaching threshold causes the influx of sodium to come into the cell, the opening of sodium voltage gated channels. Okay, that information reaches the axon hillock and causes the promotion of the action potential down through the neuron. And we talked before that the action potential is propagated all the way down the length of the axon, but in the presence of these myelin sheaths, what happens is the action potential isn't propagated in the area where there's a myelin sheath, it's just propagated in these spaces in between. So you can see that having these myelin sheaths would really help speed the action potential production down the axon. Does anyone know what the process is called when the action potential skips from here, comes right on through to here? Yeah?
Is that saltatory conduction?
That is correct. This is saltatory conduction. It's nothing more than the jumping of the action potential past the myelin sheath. So that's saltatory conduction. Okay, and saltatory conduction means that that action potential is just not happening in places where there's a myelin sheath. It just appears before and after the myelin sheath. And then these spaces in between the myelin sheath have a special name. So anywhere there's a space in between the myelin sheath, it has a name. It's called the node of Ranvier. Yes, very fancy. Those are called nodes of Ranvier. Okay, so saltatory conduction is the process of the action potential jumping right on past the myelinated segments. Node of Ranvier is that little space in between each of the myelin segments. Questions? Should be pretty straightforward. Now, moving on down here, we have CNS and PNS. Let's start over here with the CNS side and talk about does anyone know the name of the myelinating or demyelinating disorder that's found in the central nervous system? A lot of people have it, some people, anybody?
MS?
MS, yeah, multiple sclerosis. So here's going to be in the CNS, the demyelinating disorder that's fairly common these days is called multiple sclerosis. And this is actually an autoimmune disorder where the body actually attacks and destroys the oligodendrocytes. So let's say it causes multiple sclerosis is a demyelination of specifically the oligodendrocytes. These are found in the CNS. Okay. And you should know that for multiple sclerosis, the signs and symptoms are going to be variable from person to person. Okay, no two people are going to have the same things, and they tend to be more symptoms, meaning things that people are complaining about, whether as opposed to something I can see like blood pressure or heart rate or something like that. So things like fatigue. Fatigue is going to be really common. Visual disturbances, yes, Ellie?
MS is really temperature dependent, isn't it?
No, it has to do with the myelin.
I understand.
So, yeah.
The
So essentially, if patients with MS get hot, they have what's called a pseudo-exacerbation, but it doesn't increase the rate of the disease. Okay, so they just have more symptoms. Yeah, they have more symptoms. Okay, so signs and symptoms are going to be variable, and for MS, this is going to be a lifelong problem for this person. In other words, once you're diagnosed with MS, you're going to have it for life. It's an autoimmune condition that we're working on lots of different treatments for it, but it's going to keep getting worse over time. Okay, there's one demyelinating disorder in the PNS. Actually, there's two. Does anybody know the demyelinating disorder in the PNS? It's going to be Guillain-Barré. Guillain-Barré syndrome. Okay, and Guillain-Barré syndrome is also autoimmune. Okay. Guillian. Barre is Barre. It's got a little apostrophe thing. And this is a demyelination problem, specifically in the PNS. So what's the name of the cell that's going to be affected with Guillain-Barré? Yeah, that's your Schwann cell is going to have problems here. So your immune system is going to attack your Schwann cells. And then let's say 80% of patients are going to have a full recovery, but it's going to take about 2 years. Okay, that's what you need to know. And then the other one, there's a weird one that's not autoimmune, but it's genetic. It's called Charcot, Charcot, Marie tooth. And this one's genetic, so you're born with it. Okay. Signs and symptoms aren't present when you're born. They take a while to appear, but essentially you're going to have decreased lower extremity, specifically strength and sensation. Charcot-Marie-Tooth, the genetic disorder is specific for a protein called connexin that's an important part of myelin. And without connexin, the myelin degrades over time, but it does take a while. A lot of stuff today. Wednesday we have our first cases. Brain cases are fun. I like them. Hopefully you'll like them too. Yes, Gabby.
So last